Welcome again, guys. We are talking about chicken embryology, and previously we have talked about the cleavage in chicken. Now, in this video, we'll be talking about the gastrulation. Gastrulation in chick. Okay. So, for gastrulation in chick, what we know that gastrulation, we have already talked about gastrulation completely. We have talked about it details in previous video. So we won't be talking about it in detail now. So only what we'll be talking about here, we'll be talking about the basic process and the uniqueness of this chicken embryo formation and chicken gastrulation here, right? So after the cleavage is done, remember cleavage begins from zygote and first furrow, second furrow formation, and finally we get 32 cells. Now the 32 cell stage is simply termed as blastocyte. So we get blastocyte here. So once we get blastocyte, now it will be matured through the gastrulation process, and we know gastrulation is simply a modification of cells. Organizers start to occur, and finally cells are being matured, and those totipotent cells are finally getting some meaning of their life to produce a specific kind of cells with specific functions, right? That's what gastrulation is all about. Now for this process for chickens, if we start to talk about it, let's say this is the chicken egg. Now in this chicken egg, what we are seeing that at the very beginning, this is the yolk sac region with the yellow color. We'll be drawing the yolk sac. So this is the yolk sac section. Now what is going on at the very beginning process, the terminal cells, this outer layer cells start to occur. So at the very beginning, cells start to divide, and the cell division start to occur onto the top layer of this yolk sac. So inside this yolk sac we are having yolk, don't need to worry about it. So what is going on here? Cells start to form. So cells start to form onto the surface of this yolk sac. So here we come the yolk sac region. Cells, let's say these are the cells start to form inside this yolk sac. So still cells start to form like this. So as the cells are growing and developing, this is the first layer of cells that are going to form at the very inner layer of cells, right? Then after certain times later, a new layer of cells start to emerge onto this first layer of cells, right? So let's say this is the first layer of cells, and the new layer of cells start to emerge there. Throughout this place, a new layer of cells start to form. Now during the formation of this new layer of cells, remember this particular yolk sac or yolk region start to deplete, right? So as this new layer or second layer start to form, which is going to be the outermost layer of this shell of this egg, which is also called the ectoderm, is start to form, and during this process depletion of this yolk sac start to occur. So what is going on there? In this case, they are kind of coming inside like this, and also this outer layer is coming outside like this. During this process, this yolk sac is going very less, it's cutting down, right? And what they form sooner, if we imagine this inside a layer like this, what they form sooner, they form a pore outside. This is called blastopore, right? And hollow cylinder was inside, which is called blastocele. So blastopore start to occur through this part, through this pore actually. Through this pore, cells are kind of forming invagination inside, and what we see, two different types of layers start to form. The first layer that was start to form onto the surface of yolk sac are termed as these are the layer. They are termed as let me light they're termed as endoderm, and the second layer, which is start to form onto the surface of endoderm, are called ectoderm. But where does the middle layer come from? Because we know that there's obviously middle layer. Now after the formation or the invagination is going on by this ectoderm and endoderm, it is going on. During this process, another new layer of cells start to form, and they start to take this over opposite direction through this layer, and this new layer of cells again from the blastocyte, is moving at this orientation, in the anti-clockwise orientation, and sooner it will reach in between these first two layers, and it is termed as mesoderm. Mesoderm. So after ultimately completion of all these stages, what we'll be seeing one ectoderm completely outside, one mesoderm, sorry, so ectoderm, endoderm inside, and one mesoderm that we've seen in between ectoderm and endoderm, right? And inside that we'll be having that small yolk sac region there, and the developing embryo will be attached to the that developing embryo will be there in the allantois, right? So a new formation will be there, which is called allantois. We'll be talking about it later. It will look something like this, where the developing embryo will grow allantois, right? So then this is the yolk sac. Let me write, this is the yolk sac, and then these are the organizing system. Now soon they form this particular system. The rest of the things remains kind of same, because the ectoderm produces nervous system, parts of eye, the feather, beak, claws, skin, all of these things coming from ectoderm. Now endoderm produces the digestive system, remember digestive system, and different secretory organs like liver and all these things. Now this final layer, or this mesoderm, the interlayer of the ectoderm and endoderm, the mesodermal layer, it produces circulatory system, skeleton system, muscle system, reproductive organs, and excretory system in a bit. So in a sense, production process from different layers kind of remains the same for one type of embryology like chicken with frog or sea urchin, all of them kind of same, but the process of cleavage and gastrulation are hugely varied, right? So this is in a nutshell the process of gastrulation for chicken, and I hope that's helpful, guys. Thank you.